Hello, everyone. My name is Stephanie Bobadilla-Regalado, and I was an intern of the Gregory Lab at the University of Pennsylvania this summer. Today, I will be talking to you about some of the unique upregulated genes in the female reproductive tract, specifically the immunoglobulin genes. So let me tell you a little bit more about our project. Our ultimate goal was to learn what cell types are present in the four main organs of the female reproductive system. And once we know that, then we can spatially map out where these cells are located within the tissue. So let's talk about how we are conducting the investigation. The data that I'm going to present today comes from somebody who is undergoing gender redefinition treatment, and they were going through high levels of testosterone. And so the O'Neill Laboratory at the University of Pennsylvania did our tissue dissociation, and they cutted the cervix into 22 layers, and they performed single cell suspension, which is just a culture where the cells are allowed to multiply, so that we can obtain high resolutions of what cells are located in each splice. So then here comes the fun part, which is the single cell RNA sequencing. And this approach provides the expression profiles of individual cells. And so within the data, you can identify the different cell types. And according to the Cystic Fibrosis Center at Stanford, the human genome is estimated to contain approximately 80,000 to 100,000 genes. So you can imagine how much data we get from this method. But a common method that is used with this single cell sequencing is dimensionality reduction by HuBMAP, which is uniform manifold approximation and projection. So this simply refers to the process of reducing the number of attributes in a data set. And so we do lose some of the variability from the original data set, but it helps reduce the noise. And at the end, there are more pros and cons with this method. And so I'll talk more about HuBMAP in our next slide. But just to finish this one off, after we create the HuBMAP plot, you can spatially map out where these cells are in relation to the organ's tissues by using FISH. And so FISH is a method for detecting particular RNA molecules within the cells via fluorescence microscopy. And basically, you just attach a fluorescent dye to a piece of DNA, and then you target the RNA sequence within the cell, and that's how you detect the location of individual RNA molecules. So that's the overall method section. So now I'll move on to the data. So like I mentioned before, all of our data, after all of the data correction, at the end, you get a plot like this one, which is after HuBMAP. Each dot represents a single cell, and the dots or cells that are clustered together are more similar to each other. So like these ones over here, or these ones over here. And then you can identify lots of different cell types by looking at the genes within those clusters. And so for this presentation, I chose to focus on cluster 12, which is just this one right at the bottom here, isolated. And I wish I could go over each cluster and tell you the main genes found in them, but due to the short amount of presentation time, I will only be going over the top 5 upregulated genes in cluster 12 as seen in this table. And so the p-values for all of these are very significant. IgDH is a gene in cluster 12 and is upregulated 9.72 times, which makes it the most common gene found in that cluster. It has a p-value of 5.17 times 10 to the negative 17. And you can note that all the upregulated genes in cluster 12 are immunoglobulin genes, or otherwise known as antibodies. And so this G chain right there, that doesn't start with Ig, but it is the joining chain of multimeric of 2 antibodies. So it still falls into that category. And so there are 3 main antibodies, and yes, there are a total of 5, but most commonly there are 3, and those are IgA, IgG, and IgM. IgAs protect body surfaces that are exposed to the environment, so these are found mostly in the nose, ears, eyes, and other parts. IgGs are found in all body fluids, and it controls infection of body tissues. These are the ones that protect you against an infection by remembering what germs you've been exposed to in the past. And then there are IgMs, and these are the largest antibodies, and they are the first to respond to an infection. And so how do these antibodies have to do with the female reproductive system? Well, in a study done by Bredzovic et al., the levels of these three antibodies are dependent on the menstrual cycle and are influenced by hormones. And so this table you see here in this slide is done by Gonzalez et al., where they compare the immunoglobulin levels by sex. And so you can see that females have less IgAs, but they have more IgGs and IgMs in serum. And so now in seminal fluid, Bredzovic et al. Concluded that IgGs and IgAs are most prevalent in men. And so based on the published data and our data, we do expect to see these immune cells in the female reproductive tract, which is why we see them in cluster 12. So with a little bit more time, we can analyze if our donor is transitioning to a collection of male-like genes. And so what is next for our lab? Well, this was the data for only the cervix, so we still need to move on to the next three organs of the female reproductive tract and do exactly what we just did for this, but for those organs. And then we can spatially map this using the FISH method mentioned earlier so that you can detect the location of individual RNA molecules within the tissue and or the organ. And like I mentioned before, this donor was undergoing hormone treatment, so there can be a comparative analysis with no treatment and determine if there are different cells that dominate each cluster. And so none of this would have been possible without Dr. Gregory and Ron Guo from the University of Pennsylvania. Ron was the one who made the libraries. Thank you to the O'Neill Lab for taking the samples and conducting the tissue dissociation. And finally, I would like to thank the NIH and HuBMAP for funding this project and giving me the opportunity to be a part of this. And I'm happy to take any questions at this time.